The next figure we'll talk about covers this method that's called microarray, or sometimes scientists call it DNA microarray. And the purpose of DNA microarray is to compare gene expression between two cell types. And just to get us started on that, we could take a cell from the brain and a cell from the pancreas. And both have the same DNA. So that's an important concept that we learned about. And the concept is the final theme of our course. It's called totipotency. You know, in theory, these guys have, since they have all the DNA, in theory they could be used to make a whole organism totally potent. And then it's connected, totipotency, often there's another term that's used, they are said to have genomic equivalence. And so they have this potential, yet the cells are different. How are they different? And that brings up this second part of our final theme, and that is the concept of gene expression. And we learned a lot about that in chapters 17 and 18. And we learned about the organization of the gene into the promoter and the transcription unit, and how the promoter regulates the transcription unit. And DNA microarray is following up on this idea that, yeah, each cell has all of the genes, but is only using a subset of that information. And so DNA microarray compares gene expression between any two cell types. And it could be any two cell types. So you could have a normal cell, right? And you can have a cancer cell. And those cells are different because they're expressing different sets of genes. And so let's take a look at those genes. Or you could have a normal cell, and you could have a stem cell. Those are really interesting cells, and we're going to actually talk about an example on those cells. Then any other cell. You could have a cell with no medicine, and a cell that's been treated with medicine, to see how that medicine affects gene expression, what genes are switched on and off. You could have a cell with no hormone, and then one that's exposed to a hormone, to see how the cell responds to that hormone. And so DNA microarray has, another one example where it's 1,001 different applications. And here's how it works. So take a tissue sample from one cell type. And the problem with this figure is it just shows one cell type, and it'd be more effective if it showed two cell types. So let's just say we'll use brain in one and pancreas in the other. And so the RNA is purified. That's the genes that are being expressed. So we've got DNA, and the DNA represents all the genes, and that's found the same in both. So we're not interested in studying the DNA, we'd like to study the RNA. So let's purify the mRNA, and then the RNA is tagged. And this RNA is tagged with a yellow label, and then this one will be tagged with a different one, let's say red. And then the DNA is hybridized to a target. And so we need to just talk about that a little bit. In PCR, we talked about how the DNA could be unzipped, and then when it's cooled, the primers will zip or anneal. And that's the scientific term for the zipping together of two strands, is annealing. And so what's done is take these labeled strands, mix them, and incubate them in the presence of this target. And what the target has is it has a little piece of DNA from each gene. And so one of these would be the insulin gene, and one would be the hemoglobin gene, and one would be ATP synthase gene, and LDL receptor gene. And we have about 25,000, 30,000 different genes in us. And so there would be all these little dots for a human genome. And so what we're going to do is we're going to study all the cells, all the genes at the same time. We might be interested in the insulin gene, but at the same time, we're just going to study them all. And at the proper temperature that will allow the DNAs to zip together, this RNA is mixed with the DNA and gets zipped together. And so any genes that are expressed in the brain will be tagged with a yellow on their RNA, and then those will glow yellow after the annealing is done. And on the other hand, any genes that are expressed in the pancreas will glow red. So let me just erase this and go like this. So there'll be some genes like that. They glow, some glow red, some glow yellow, many glow both. And it creates kind of an orange color because they're expressed in both cell types. And then what's done is, again, a spectrophotometer is going to look at each one of these spots and measure the amount of fluorescence. And if it detects red fluorescence, then this is a cell, the gene that's made in the pancreatic cell, like that could be insulin, like that. And so that's the DNA microarray method. It allows us to look at comparison between DNA sequences in any cell type. And this is just a figure I found on the web. But again, we got one figure in the book, and you can just Google an image of any of these on the web, and it'll show you and help you understand it. So here it's saying, oh, let's take a normal and a cancerous cell, grind them up. We'll tag the normal cell's RNAs with green, the tumorous cell RNAs with red, and anneal them to our microarray target grid. And we'll see some genes are not expressed at all in either cell type. So they're human genes, but they might have been used maybe in the embryo, like that, or in the brain, and they're just not expressed in this tissue. So some don't glow at all, and then some glow. Oh, actually, what this is showing, depicting is, I think, is if they glow both red and green together, you get kind of this yellowish orangish color. Yeah, sorry. But there would be, it should show that, there would be some that just aren't glowing at all because they're not expressed. That's the case in any cell. No cell expresses all the genes. And then some would glow red and green, like that. So that's the DNA microarray purpose, and that's what the work looks like. Here's just another figure I grabbed from the book, just showing you this idea of zipping together with the target. So this is just one of those spots on that array, and this would be one gene represented. But on that spot, there are, it says, millions of DNA strands, and they're all the same sequence. They're all that same gene. And then when the incubation is done, these tagged target RNAs will zip up, will anneal in a base pair specific way to the targets, and then the excess is washed away and the fluorescence measured. And so that's how DNA microarray works. And the end of this one.